Okay, so paper chromatography, we have different type of chromatography. We have gas chromatography. With help of gas, they separate molecules. We have high pressure liquid chromatography. With help of liquids, they separate molecules. Now this one that you did here in the lab, it was on a piece of paper. On a paper, it was a special type of paper. You separated different type of molecules, okay? If I ask you, it was a question in your lab handout, if I ask you which one on your paper, for example, you put the drop here, you remember that? Okay, which one would be the heaviest one and which one would be the lightest one? Out of those pigments you separated, chlorophyll A, right? Chlorophyll B, carotene, and xanthophyll. Do you remember those? So which one, chlorophyll B, A, C, or xanthophyll, which one is the heaviest one?
B.
B. B did not travel far, so it's the heaviest one. Which one is the lightest one? Xanthophyll. Okay? Xanthophyll. Xanthophyll was the lightest one. Am I making some sense? Everybody, how things go? And this is what you did on Tuesday, last time, Wednesday. Okay, so anyhow, you had chlorophyll A, which participated in photosynthesis directly. I gave you an example. You have Erlenmeyer flask. Remember that? Which one? Water goes faster through a straw or through a funnel. Okay, so chlorophyll A and other chlorophylls act like a funnel. Chlorophyll B, xanthophyll, carotene, they all collect light and they give it to chlorophyll A. I'm going to make you some sense, everybody, how that photosynthesis works. So anyhow, and here are different wavelengths and chlorophyll and so on and so forth. Most activity occurs around here, 680, 700, which that's what we are going to talk about next time. This is the structure of chlorophyll, and I showed it to you last time, and I said, okay, we will go over it. Here is what is happening as far as the thylakoid membrane, and here are the protons are being transmitted, and so on and so forth. But let's go over it. There are two types of photosystem in thylakoid membrane, photosystem 2 and photosystem 1. Photosystem 2 works with best with 680. If you go back here to 680 right here, around here, that it works the best. Photosystem 2, I'm talking about chlorophyll A. All of this is about chlorophyll A. Okay, all of this is about chlorophyll A, and system 2 functions first, numbers, reflects order of discovery and best absorbing A wavelength at 680 nanometer. The reaction center chlorophyll A of PS2 is called 680. Okay, the system 1, photosystem 1, it works best at 700 nanometer. I showed you on the prism light where 700 was. Should I go back again? Oh, I showed it to you at 680 and now this is 700. Okay, so of course this is pretty much we already talked about. You already know that light comes into thylakoid and then water comes into thylakoid. Water is oxygen is released from this reaction, and then of course we will talk about what is happening in thylakoid. ATP and NADPH is synthesized. When ATP and NADPH synthesized, go through the Calvin cycle, put all of this carbon fixation. I did write it on the board last time. Let me write it again. It's called carbon fixation. For carbon fixation, what does that mean? Carbon fixation, when you are putting carbon dioxide and carbon dioxide, eventually you're getting sugar molecule, which is your C6H12O6. Am I making some sense? That is carbon fixation. For carbon fixation, of course, you need ATP and NADPH. You need ATP and NADPH. When carbons are being put together and make sugar molecules, then those ATP and NADPH turn into NADP and ADP. I hope I'm making some sense. Okay. Here again, light is coming into photosystem 1, generating some, stimulating some pigments, and those pigments, of course, they make electrons, and those electrons, primary acceptors. Water comes in, and water breaks down to oxygen, and then, of course, those electrons, again, prop up those pigments. And then it goes through electron transport chain of PQ, cytochrome complex PC, and make ATP, of course, and then it goes to system 1, P700, that's P600, and then of course light will stimulate. The best analogy, and at the end you get NADPH is synthesized. The best analogy I have, your textbook has, is for photosystem 1 and photosystem 2. Right here, the man hits, there is a ball right here, he hits it with a proton, and the ball jumps up, he grabs it, she grabs it, and pull it down, and then a slope, and then it goes through the wheels, and it goes down here, stands right here, and the man, again, another man, hit it, whatever you call it, it goes up here, he catches it and put it in a bucket. And bucket is made, NADPH is made right here, ATP is being made. So it's an analogy your textbook gave. That's what happens with photosystem 2 and photosystem 1. Photosystem 2 works, this is the number that I'm hoping you remember for Wednesday. Photosystem 2 works with 680 nanometer, the wavelength of light, 680, and photosystem 1 works with 700. And it's all chlorophyll A. It's all happening in chlorophyll A. Okay. Cyclic electron flow. The cyclic electron flow, electrons cycle, break from FP to PSI reactions center. Cyclic electron flow uses only photosystem 1 and produces ATP, but not NADPH. No oxygen is released during this time. And the next thing your textbook is doing, that is comparing the chemoosmosis of chloroplasts and mitochondria. Okay, so the chloroplasts and mitochondria generated ATP by chemoosmosis, but use different sources of energy. What do you think? I should have covered this one. Maybe I should stand in front of it. In mitochondria, in chloroplast, you're making ATP by what? What is the source of energy? What do you think? Huh? Light. Light. In mitochondria, you're making ATP by what? By glucose. Okay, so the other way around. Chloroplast by light and mitochondria by glucose. Mitochondria transfer chemical energy from food, yes, glucose, and eventually, of course, the glucose breaks down to pyruvate and makes ATP. Chloroplast transforms light energy into chemical energy of ATP. Spatial organization of chemoosmosis between chloroplast and mitochondria also shows a similarity. In mitochondria, protons are pumped to the intermembrane space and drive ATP synthesis as they diffuse back into the mitochondria matrix. In chloroplast, protons are pumped into the thylakoid space and ATP synthesis happens right here. Okay, let's go over this a little bit. You're comparing mitochondria and chloroplast. Mitochondria, chloroplast. Mitochondria has the outer membrane, the inner membrane, the cristae, and matrix. Those are the main parts of mitochondria, right? This is the matrix. All of that inside layer of mitochondria is called matrix. Here in chloroplast, you have outer membrane, inner membrane, thylakoid, and stroma. Remember that? This is stroma, this is matrix, right? Am I making some sense, everybody? Okay, now let's see what happens. So, you have in matrix, this one, in matrix as a mitochondria, you have electron transport chain. In mitochondria, So what happens? ADP becomes ATP by protons. You know the protons are accumulated here, chemiosmosis, and they go through ATPase, and they eventually make ATP. You remember that from last chapter, from cellular respiration. Now, what happens here in chloroplast? Same thing again. A lot of what happens, this is the thylakoid, and of course the ATPase or ATP synthase is right here. And then you have a lot of hydrogens here. These hydrogens go through here and turn the ADP to ATP. Okay, so the level, the organization of it is different, but eventually ATP formation is from those hydrogen pumps, from hydrogens, come from hydrogen. Hydrogen turns, cause conversion of ADP plus phosphate to ATP, and then of course this is a stroma right here, that's happening, stroma, but this molecule ATPase, ATP synthase, I call it ATPase, same thing, is right here, where the laser is, and over there is right there. Okay, I hope I'm making some sense. Okay, ATP and NADPH are produced on the side facing the stroma where the Calvin cycle takes place. In summary, light reactions generates ATP and increase the potential energy of electrons by moving them from water to NADPH. Yes. Here we go. Again, what was the summary here? This is a summary of course, comparing mitochondria to chloroplast. This is a summary of what happens here. The next few slides, it talks about it in more details. I'm not worried about it. I just put it out there. If you guys are interested, you can study it. I'm not going to ask any detailed questions here. What happens for hydrogens and so on and so forth. I'm not worried about it. So I don't know if you want to study it for your own. I put it up here. What happens? Okay, the Calvin cycles, but again, these write-ups are important. The write-ups are important. These 3 pictures or 4 pictures I'm not too crazy about. There is more detail than what I'm expecting from you guys. Okay. Calvin cycle, like citric acid cycle, regenerates its starting materials from molecules and and leave the cycle. The cycle builds sugar from smaller molecules by using ATP and reducing power of electrons carried by NADPH. So, G3P, you remember before, you remember I talked about carbon fixation? You put the carbons together in order to form the sugar molecules. But before we start putting the carbons together, in the chloroplast, there is a molecule called G3P. It means that molecule already has 3, you saw it in the video also, that molecule in the chloroplast already has 3 carbons. Okay? Then another 3 carbons, I'm going to draw it in different color, hopefully. So I can, it means radioactive. Another 3 carbons comes and then attached to it. So this portion right here, we're calling it a carbon fixation. Carbon fixation. And of course, for adding these carbons to this molecule, to G3P, that carbon, and that carbon, you need ATP and NADPH. Do I make sense? So it's not 6 carbon dioxides come together and form a glucose molecule. No, there is already a 3 carbon molecule in the chloroplast, and then another carbon comes and attached to it. It becomes 4 carbon. Another carbon comes attached to it. It becomes 5 carbon. Another carbon comes and attached to it. It becomes 6 carbon, and that 6 carbon leaves the chloroplast and goes to different parts of the plant, or stays in the cell and being stored in mitochondria in a vacuum. Okay, so that's pretty much what carbon fixation means. Had 3 phases: carbon fixation, catalyzed, Rubisco. Do not worry about that, and reduction and regeneration of the carbon acceptors, Rubisco. Here it is again, going over the same thing. You've seen this slide 100 times so far, I guess. Sugar production in C4 plants occurs in 3 steps. Production of 4 carbon. So what am I saying here? That plants, we have 3 different type of plants. Maybe I should erase this. No, doesn't like that. Doesn't like that either. We have 3 different type of plants. And I'll run it by you guys a little bit. The 3 type of plant are C3 plant, C4 plant, and CAM plants. C3 plants, I just talked about it. Most plants are C3 plant. Most plants are C3 plants. C4 and CAM are, you will see, and I'll show you some pictures. Okay, what they are. C3, the initiation molecule has 3 carbons, and then 3 other carbons come attached to it, becomes a glucose molecule. C4 plants, the initiation molecule has 4 carbons, and 2 carbons come and attach to it, becomes a 6 carbon molecule, glucose, right? And a CAM plant, we call it crassolucent acid metabolism, which is a little bit different. It uses a crassolucent acid as an initiator, and the carbons come and attach to it. Okay, so let's go. Again, these are fine. Here is again talks about what happens in C4 plants in detail. Again, I'm not too crazy about it. I'm not too worried about it. But what I am worried about is CAM plants. Right here, some plants, including succulent, uses crassolucent acid metabolism to fix carbon. CAM plant open their stomata at night, incorporating carbon dioxide into organic molecules. Okay, so these guys open up their stomata at night. And the stomata, you know this, what stomata is. I brought you a slide the other day. Today, I'm hoping you will look at the slide today again, and these openings are called stomata. You remember that? This is called stomata, the opening, the pores on the leaf, called stomata. So they open up stomata at night, incorporating carbon dioxide into the organic molecule. Stomata closes during day, and carbon dioxide is released from organic acids and used in Calvin cycle. Here they are talking about like pineapple is a CAM plant. Sugar cane, these 2 are CAM plant. Okay, and then they are incorporating these into their C3 plants. Are plants with carbon dioxide directly from the air: soybean, rice, wheat, oats. I gave you several examples. Most plants are C3 plants, and I gave you some examples: soybean, rice, wheat, oats. On dry days, stomata closes to reduce water loss. C4 plants have its stomata closed at most of the time to save water. They use the enzyme to fix a 4-carbon compound instead of a 3-carbon compound. These plants cannot fix oxygen. So C4 plants do not do oxygen fixation. They do not release oxygen into the atmosphere. C4 plants. Okay. Did I give you any examples of C4 plants? Is corn, sorghum, and sugarcane. Those are examples of C4 plants. CAM plants open up their stomata, admitting CO2 only at night. Calvin cycle occurs during the day. CAM is crassolucent acid metabolism. Example: pineapple and many cacti and succulent plants such as juicy plants and jade plants. These are all. So what I'm hoping you guys would know, as far as these 3, you should know examples of C3, C4, and CAM. And then just generally, I gave you some examples right here, these last two PowerPoints, I gave you some examples. Okay, these two, I gave you some examples. And this, you should know C3 plant, most plants are C3 plants, and then the initiation molecule is a 3-carbon molecule. This one is a 4-molecule carbon, and 2 carbons come attached to it. This one, C3, 3-molecule carbon, and then 3 carbon, one by one, come and attach to it, becomes a 6-carbon. And CAM is crassolucent acid metabolism. Some might ask you that. Okay. And then importance of photosynthesis, the review. Energy entering chloroplast as sunlight gets stored as chemical energy in organic compound. Sugar made in chloroplast supplies chemical energy in carbon skeleton synthesis, the organic molecules. Okay, these are again, I'm not too crazy about this one. Here it gives you the circulation of carbon and water, and so on and so forth in plants, and the greenhouse effect. We need to talk about greenhouse effect. Have you heard of greenhouse effect? What is happening to the Mother Earth? What is greenhouse effect? What here is the Mother Earth. And here is, if I have a blue, do I have, yes, here is the atmosphere. Is that right? You all know what I'm talking about. And then what happens, plants on the surface of the earth, most of them, most of C3 plants, they take carbon dioxide, right? And they give off oxygen. And we human animals will take that oxygen and give back carbon dioxide to the atmosphere. Oh, I should put it back this way. And you saw that before, right? Am I making some sense? However, what we are doing, we are building factories and cars. Right here, I'm going to draw the cars and other goodies on planet Earth, and these guys are putting a lot of carbon dioxide into the atmosphere. Am I making some sense? Ever than before. Before the Industrial Revolution, what happened? That balance existed, so nothing was happening on the surface of the Earth. But now there is a lot of course, methane gas, CH4, sorry. CH4, methane, also accumulated on the surface of the earth. Nitrogen oxide, another one. So there are lots of, well, these are the 4 main gases are accumulating on the surface of the earth. And then what you have, here we go. You have sun. Sun sends rays, of course, for photosynthesis. Am I making some sense? Not only light comes with the sun, also heat is coming with it too, right? Not only light comes from sun, also heat comes from sun as well. So what happens when light comes and that heat is coming too? All of these gases on the atmosphere, these gases on the atmosphere do not allow the heat to go back. It reflects back. When heat comes, when heat is coming from sun, it comes to the earth and it reflects back. It's supposed to. It did before Industrial Revolution. Am I making some sense? But now the heat is coming, but it's getting trapped right here. It's not reflecting back out of the earth, atmosphere. And that's why Earth is warming up. That's why the glaciers are melting. Trump is surprised. Are we doing this to Mother Earth? Yes, Trump, we're doing it. greenhouse effect. Some gases such as carbon dioxide, nitrous oxide, and methane can trap the heat from the sun to warm the earth, similar to glasses on a greenhouse. Have you ever guys gone to a greenhouse during winter time, during this time of the year? Huh? Have you ever gone, you haven't gone to a greenhouse. If you go there to a greenhouse, and you know what the greenhouse is, they make glass. The greenhouse has a short wall up to here, but the rest, the ceiling, the roof, everywhere is glass. Because light comes in, perform photosynthesis, and the heat does not get out. If you go to a greenhouse, it's right now warmer than this classroom. You're freezing in here. But if it was all glass, it would be nice and warm. Am I making some sense, everybody? So heat does not leave greenhouse. Same as comparing the present condition of the Earth to a greenhouse. Right now, these gases are on the atmosphere, do not allow heat to leave the greenhouse, the Earth, the surface of the Earth. Increased use of coal, oil, gas, and wood as a fuel. Photosynthesis counteract accumulation of carbon dioxide. We are cutting our forests, so photosynthesis stops. Where do these gases come from? Increased use of oil, gas, and woods and fuels. Photosynthesis counteract the accumulation of carbon dioxide. We are cutting our forests, and photosynthesis stops. And here are the end of the material for this exam, for this chapter. Is there any question? Does anybody have any question?